All right, I'm going to go through the basics of the chemical synapse, neurotransmitters. Since this is a review, I'll be going through it fairly quickly. I will ask you some questions as we go. In this kind of format, I'm not going to wait for you to actually answer me, but the questions aren't rhetorical. So I expect you to be able to answer the kinds of questions I ask here on the final. I'm not going to ask you the same ones exactly, but that's the level of understanding I want you to have. I'm going to draw this out on the whiteboard. If you have questions as we go, please just type them in the chat window. It's easiest if you ask as we go. That way I can address them right there. All right, so when we have the neuron, neurons don't really work in isolation. We have circuits of neurons, and they're going to connect to one another and communicate, send information. Within a neuron, the communication is electrical. And so on the axon, that's where we talk about the action potential. Once the action potential hits the terminal, now somehow we have to transfer that information from one neuron to another. Several different mechanisms for doing this have evolved. There are so-called electrical synapses or gap junctions. Gap junctions are going to be actual continuations, essentially, of the axon, so that ions from this neuron will actually flow into the next. Okay, so there's physical bridges between them. These are relatively uncommon in vertebrates like us. In insects, there's lots of electrical synapses. We do have them. Your heart muscle is largely controlled by these types of connections of neurons. All right. But for psychology, what we really are going to care about is this chemical synapse. Okay, so we're going to transfer information from one neuron to another. We're going to use a chemical. The chemicals we use are the neurotransmitters. And I'm going to spend a little bit of time talking about the different categories of neurotransmitters, and then a little bit about receptors. And then I will get specifically into the neuromuscular junction and the monoamine synapse. All right, so with the neurotransmitters, obviously these are going to transmit information between neurons. There's many different classes of neurotransmitters. And so neurotransmitters are classified based on their chemistry. And so we'll talk about glutamate and GABA. Okay. These are have distinct chemical structures, obviously. Glutamate is the major excitatory neurotransmitter in the brain. GABA is a major inhibitory. In addition to these, we're also going to have acetylcholine, capital A, capital C, lowercase h. Acetylcholine is important for movement. It's how neurons control the muscles. But you also have a lot of acetylcholine in your brain. With acetylcholine, you can think Alzheimer's disease. Right? When we talk about cognition and we talk about sleep, one of the things hopefully you got was that acetylcholine being released in your cortex stimulates the cortex. It gets that cortex going. Right? With Alzheimer's disease, these neurons die fast. And so early on in Alzheimer's, It's these acetylcholine neurons in the forebrain and nucleus basalis that degenerate first. That means that the cortex isn't as aroused. It's not paying attention as much, which is why some of the early signs of Alzheimer's are, you forget where you put your keys, things like that. Right. We'll talk a little bit more about that. We also have a broad class called the monoamines. The monoamines are made from a single amino acid. So it's chemically modified, but we're going to take an amino acid and modify it. Within monoamines, we will have the indoleamines, and we'll have the catecholamines. The indoleamines, the only one we're going to talk about is 5-hydroxytryptamine, which is also known as serotonin. For the catecholamines, there are 3. We'll have dopamine, norepinephrine, and epinephrine. In terms of psychology, dopamine is hugely important. When we talk about the reward pathway, addiction, dopamine is central to those pathologies. It also plays a major role in some of the symptoms of schizophrenia. Norepinephrine is important for arousal. I don't mean sexual arousal. I mean paying attention to what's going on around you, vigilance, that type of stuff. In psychology, in my class, we largely ignore epinephrine. Epinephrine plays a much more important role in physiology, things like stress response, things like that. In terms of behavior and psychopathology, we don't know much that epinephrine plays a really key role in. As you know, there's enough in this class, so we'll focus on serotonin, dopamine, and norepinephrine. Okay, so your monoamines are indoleamines, catecholamines. Now, one thing we did talk about, and hopefully, again, you got genes in your DNA code for proteins. Okay, only proteins. If it's not a protein, there's no gene for it. And so people talk about genes for intelligence. As a behavior geneticist, I roll my eyes at that. Intelligence is not a protein. So there's no gene for intelligence. Now, there are genes that code for proteins that affect the way the brain is put together, the way it develops, and the way it functions. And if there's a problem with one of those, that can lead to deficits in brain function, which would be lower intelligence. All right, but there's no gene for intelligence. Now, the reason I'm emphasizing that, monoamines, right, are single amino acids. They're not proteins. So is there a gene for dopamine? The answer is no. There's no gene for dopamine. So how do we regulate how much dopamine we have in our brain? We don't do it through diet. Sometimes people think we do it by diet. That's only true in cases of severe malnutrition. And if you're severely malnourished, that your neurotransmitter levels can be affected. For the average person with the average diet, you get more than enough nutrients to make all the neurotransmitter you need. Here's where we're going to talk about the synthetic pathway of the catecholamines, for example. Okay, so we'll take a dietary an amino acid you get through your diet, tyrosine. Okay, tyrosine will then get converted by an enzyme called tyrosine hydroxylase. Obviously, if I'm taking the time to write these down in the review session, these are things you probably will want to know for the final. All right, tyrosine hydroxylase is abbreviated TH. Okay, so TH will convert tyrosine into something called L-DOPA. Okay, so this is just named for the chemical structure. L-DOPA gets converted by another enzyme, which you don't need to worry about, into dopamine. Dopamine gets converted by another enzyme, DBH, which again, you don't need to know, and gets converted into norepinephrine. Norepinephrine gets converted by yet another enzyme into epinephrine. Okay, so tyrosine you get from your diet. Tyrosine hydroxylase is going to convert that into L-DOPA. L-DOPA gets converted into dopamine. Now, if I am a dopamine neuron, okay, I'm going to make just dopamine. So I will have TH. I will have this enzyme that converts L-DOPA into dopamine. I want to stop here. So do you think I make this enzyme that converts dopamine into norepinephrine? If I'm a dopamine neuron, I don't make that enzyme. What if I'm a norepinephrine neuron though? Then I'm going to make TH. I'll make this enzyme. I'll make this enzyme, but I will not make this enzyme. Okay, now enzymes are proteins. Okay, so hopefully you figured out how we regulate how much dopamine you have in your brain. I have tons of tyrosine around. What I'm going to do is regulate how much of the enzymes I have. Now, it's not very efficient to regulate all the different enzymes. Okay, so it turns out tyrosine hydroxylase is the important one. That's why I want you to know the name. Okay, so I make a ton of this enzyme, a ton of this enzyme, a ton of this enzyme. Okay, so if I have a ridiculous amount of L-DOPA, I have enough enzyme that it will all get very quickly converted into dopamine. Okay, so if I have L-DOPA around, it's gonna get converted into dopamine. The way I regulate how much dopamine I have is by altering how much L-DOPA I make. All right, so let's say I want to reduce the amount of dopamine I have in my brain. What we'll do then is decrease the amount of TH. If I have less TH, less tyrosine is going to get converted into L-DOPA, which means less L-DOPA gets converted into dopamine. Okay, now the catecholamines, I want you to remember everything I've written here. Okay, so you should be able to produce this pathway. With the indolamines, serotonin, it's not tyrosine, it's tryptophan, it's different enzymes, different intermediates, but the concept is the same. Okay, that the way we regulate how much serotonin we have in the brain is by regulating the enzyme that makes it. Okay, so the names are different, but the concept's the same. Same with acetylcholine. Okay, so with acetylcholine, I'm going to move this down. With acetylcholine, we are going to have something called chat. This is choline acetyl transferase. Okay, this is the enzyme that makes acetylcholine. So if I want to increase acetylcholine levels, I don't go to GNC and buy a whole bunch of choline. That does nothing. That's truly a scam. What I do is increase the amount of choline acetyltransferase. Okay, that's going to take whatever choline I have. And again, in my normal American diet, I got tons of choline. Right? It's the enzyme that's going to determine how much. There's another enzyme called acetylcholinesterase. This is going to metabolize or breaks down acetylcholine. Okay, so with acetylcholine, not only do we regulate how much we make, we can also regulate how quickly we break it down. In this, when we talk about the neuromuscular junction, we'll talk about both of these. Right, so that's a quick overview of the neurotransmitters. Now, real quickly, what does dopamine do? Well, the effect of dopamine depends on the receptor. Okay, so there's a saying, the receptor confers biological specificity. Okay, so sometimes dopamine is excitatory. Sometimes dopamine is inhibitory. How can that be? Well, dopamine is always dopamine. The chemical structure is what makes it dopamine, right? If we alter the structure, then we don't have dopamine anymore. Okay? So if dopamine binds to a specific receptor, when that receptor is activated by dopamine, it'll have an effect on the cell. Okay, so it's the receptor that determines what happens to the cell. Sometimes it's excitatory, sometimes it's inhibitory, et cetera, et cetera. Okay, so the receptors are really important. When we talk about receptors, there's two broad classes. We're going to have ionotropic and metabotropic. Okay, so essentially here, neurotransmitter binds. I am writing. Is anyone else having trouble seeing it? What I'll do, if you can follow along listening, what I'll do is I can actually save my writing as a PDF, and I can put that up for you. You might want to leave and then come back real fast and see if that fixes it. But yeah, all right. So with ionotropic, the neurotransmitter binds. And when the neurotransmitter binds, ions flow through the receptor. This is key. They go through the receptor. Okay. With metabotropic, neurotransmitter binds, and we change the biochemistry of the cell. Okay, and we do that through a series of chemicals called second messengers. Now, ionotropic receptors are great because the effect is really fast. But as soon as the neurotransmitter goes away, ions don't flow anymore. The effect wears off immediately. Okay, so ionotropic, rapid, but transient effects. Okay, so that's good for something like muscular contraction. I don't want to walk around like this all day with my muscle contracted, right? I want it to be able to relax. So I want something that doesn't last very long. If I'm studying biopsychology, I want that to stick around, right? So here we want something that may be slower, but long lasting. So ionotropic receptors are important for sensation, perception, movement, things like that. When we talk about psychology per se, if we're talking about depression, or we're talking about PTSD, or we're talking about schizophrenia, we're talking about addiction, right? The more complex behaviors, almost all of those rely heavily on metabotropic receptors, right? So within metabotropic receptors, there's a family called GPCRs, right? That stands for G protein coupled receptor. All that is telling you is what the second messenger is. Okay, so there's hundreds of second messengers. GPCR just means the second messenger is a G protein. I know that probably doesn't mean much to you, but if someone tells you a receptor is a GPCR, you should know that that means it's a metabotropic receptor and that it's an important family for psychology. And so sometimes you'll learn that drugs alter GPCRs. Those are drugs that tend to be important for psychology. Right? In this class, you don't need to memorize beyond what I'm telling you. But the reason I want you to know what a GPCR is because they are important. Okay. Now I'm going to move on to two different types of synapses. First, I'm going to talk about the neuromuscular junction. Okay, so this is the junction, the connection between the neuron and the muscle. Normally, we're talking about neuron to neuron. The neuromuscular junction uses acetylcholine. The neuromuscular junction is probably one of the most accessible synapses. If I want to study interneurons connecting to each other in the brain, right, that's hard to get to. Neuromuscular junction is actually very easy for me to get to. So I can study it much more easily. This is also the first connection, the first synapse that was described, right? Now, although the second cell is a muscle cell, it's going to act, it's going to respond very similarly to the way a neuron would. All right, so here we have the terminal. Here we'll have the second cell. In this case, it's going to be muscle. All right. This area here is called the synaptic cleft. Okay, that's the space between these. Now, within this region, we are going to have a bunch of specializations, right? The connection between cells doesn't happen any old place. We need a bunch of specializations, and I'm going to draw some of those in a second, right? This whole thing is basically called the synapse. Okay, so the terminal, the synaptic cleft, and the second cell. Now I'm going to use the right terms here. The cell before the cleft is called the presynapse, synaptic cell. Okay, so this will be the presynaptic terminal. This will be the postsynaptic cell. And in this case, it's going to be muscle. All right, so this is the axon here. Here is the terminal. We're going to have a special channel called the voltage gated calcium channel. Within the terminal, I'm only going to draw a few, but you'll have lots of spheres filled with neurotransmitter. Okay, at the neuromuscular junction, if it's a neuromuscular junction, you know that these chemicals are acetylcholine. Okay, so this I can only draw in 2 dimensions, but these are really spheres. Those are the synaptic vesicles. You have vesicles all over your body. That's why we call them synaptic vesicles, so you know where they are. In the postsynaptic cell, you'll have receptors. Okay, what I've drawn here is pretty much going to be true of every synapse. Okay, we got the axon, we've got voltage gated calcium channels, we have transmitter filled vesicles, we'll have receptors. Okay, all this is the same at pretty much every single synapse. Now at the neuromuscular junction, these receptors are ionotropic. Okay, and these are the nicotinic acetylcholine receptors. Okay. Now, these receptors are going to be ion channels for sodium and potassium. Okay. So monovalent cations is the fancy way. Monovalent means it's +1, or I should say cation means it's positively charged. Monovalent means it's one charge. And so monovalent cation is +1. So sodium and potassium can flow through here. In addition, we're going to have something called acetylcholine esterase. Okay, so these dots represent that enzyme that breaks down acetylcholine. Right, so these are the structures, the basic structures. Now we'll go through what happens kind of step by step. So first, the action potential comes down. Right, so we have our action potential propagated down the axon. When we have depolarization of these voltage-gated calcium channels, the voltage-gated calcium channels open. When those open up now, calcium can flow. It turns out there's a higher concentration of calcium outside than inside. So calcium's going to flow in through those channels into the cell. Okay, so we get calcium influx. Now, when the calcium comes in, it's going to immediately interact with proteins. Okay, so it doesn't really affect the charge here. Instead, what it does is starts a cascade of biochemical events. Okay, so I'm just going to say calcium starts biochemical events. The result of those biochemical events is that these vesicles are then going to fuse with the membrane. And when they fuse, they release neurotransmitter. Okay, so synaptic vesicles fuse with the membrane. Neurotransmitter, in this case acetylcholine, is released. It's going to flow down its concentration gradient. Now, let's follow one acetylcholine molecule. We'll follow this. Okay, so what it's going to do is it's just going to kind of move around. If it goes here and interacts with the nicotinic acetylcholine receptor, with the receptor, it's going to open it up. Okay, now when this receptor is open, when acetylcholine is bound to it, sodium is going to flow in and potassium will flow out. Okay, now if you remember from the action potential, there's a point in the action potential near the peak where sodium and potassium are both flowing. Sodium flowing in is much, much quicker than potassium flowing out. Okay, so when sodium flows in, okay, sodium wins, and that's going to depolarize my cell. Okay, so we get a depolarization here. Now, in this class, we're not going to go through the physiology. The physiology is very well known. All I want you to know is when muscle is depolarized, we get contraction. Okay, so depolarization equals muscular contraction. All right, so my brain sends a signal down through the circuit, eventually down through the spinal cord to the motor neuron. The motor neuron is going to send an action potential to the muscle. Okay, when we hit the terminal, I have a depolarization. Voltage-gated calcium channels are going to open up. Calcium comes into the cell. My synaptic vesicles fuse and acetylcholine is released. Acetylcholine is going to sort of wander around a little bit until it comes upon a nicotinic acetylcholine receptor. When it binds to the nicotinic acetylcholine receptor, sodium's going to flow in, my muscle's depolarized, and I get a contraction. All right. Now, obviously, that's only half the story. You can't walk around with your muscles contracted forever. You would die, right? Your heart contracts and stays that way. Well, that's not compatible with life. All right, so we have to do something after this. All right, so acetylcholine doesn't bind permanently with the receptor. Okay, it's going to kind of shake hands, and then it's going to move on, and it's going to shake hands with another one. All right, now we don't want this to keep going on forever. Somehow we have to get rid of this depolarization so my muscle can relax. The way your body does that is by getting rid of all the acetylcholine in the synapse. In the neuromuscular junction, that acetylcholine will eventually interact with acetylcholinesterase. Okay. So ACH-ACE metabolizes acetylcholine. When that happens, channels close, depolarization goes away, and I get relaxation of the muscle. Okay. So let's test your knowledge here. There is a drug, a poison called curare. You've probably heard of this. Curare blocks the nicotinic acetylcholine receptor. So what's the effect of curare on a person? Well, we'll focus on the neuromuscular junction here, okay? So would curare have any effect on the action potential? No, nicotinic acetylcholine receptors have nothing to do with that. Okay, so the motor neuron sends an action potential down. I have a depolarization. The voltage-gated calcium channels, no effect there. So they open up, calcium comes in. My vesicles fuse and I get the release of acetylcholine. No problem there. Now, with curare, if curare is blocking the nicotinic acetylcholine receptor, now we have a problem, right? Acetylcholine can't bind. If acetylcholine can't bind, those receptors never open up. If those don't open up, sodium does not come in. If sodium does not come in, I don't get a depolarization. If I don't get a depolarization, my muscles won't contract. Okay, so curare, hopefully you figured it out on your own, would prevent muscular contraction. Okay, that's going to kill you, obviously. There's another set of drugs, toxins, poisons that target acetylcholinesterase. Right? You may have heard of sarin gas or the Novichok proteins. These are chemical weapons. Novichok was used to assassinate some people, including a former Soviet spy or Russian spy in England. Okay, what sarin and the Novichoks do is inhibit acetylcholinesterase. Okay, so what effect would those have on somebody? Action potential comes screaming down the axon, no problem. I get a depolarization. The voltage-gated calcium channels open up. Calcium comes in. That's going to cause the vesicles to fuse and release acetylcholine. Acetylcholine gets released and it's going to bind to and open up nicotinic acetylcholine receptors. That's going to cause depolarization and contraction. So my muscles are going to contract. Now, what if I don't have acetylcholinesterase? What if that's not working? Well, now that acetylcholine is going to continue to stimulate the nicotinic acetylcholine receptors. Okay, which means we're going to have a continuous depolarization and continuous contraction. Okay, so the muscles will contract and then stay contracted. Obviously, that's not compatible with life either. Okay, so this balance between nicotinic acetylcholine being released, activating these receptors, and then us breaking it down quickly is essential for life. You got to get that balance right or you're toast. With this, you can see like a RAID, the insecticide RAID. RAID targets acetylcholinesterase. Right. So I think probably everyone sprayed a bug before, right? So if you spray the bug and you watch it, what happens to the bug? Well, they kind of go crazy, right? They twitch and they twitch and they twitch. And then when they die, their legs are all curled up. Okay, that's because the muscles are all contracted. Now with RAID, how come RAID doesn't kill us? Well, in a very high concentration, it would obviously. But the acetylcholinesterase that insects have is a little bit different than ours. So RAID is geared toward really binding insect acetylcholinesterase very, very well. It doesn't bind ours nearly as well. All right, so that's the neuromuscular junction. Remember, the way we terminate the signal, the way we get rid of acetylcholine here is by breaking it down, by metabolism. The monoamine synapse is going to be different. All right, so with the monoamine synapse, we're going to have our presynaptic terminal. We'll have our voltage-gated calcium channels. We'll have synaptic vesicles full of neurotransmitter, could be dopamine, could be serotonin. We'll have our postsynaptic cell with receptors. Okay, so things here are largely going to be the same. Okay, so I have an action potential that's going to come down, open these voltage-gated calcium channels. Calcium's going to come in, initiate some events that are going to cause this vesicle to fuse and release neurotransmitter. This neurotransmitter is going to diffuse across the synaptic cleft, and it's going to activate receptors. And that'll give us an effect. Now, what that effect is, again, depends on what this receptor is. For monoamines, these are almost always metabotropic GPCR receptors. Okay, so we're going to alter the biochemistry of the cell. Delta means change biochem. Now, again, we don't want this effect going on forever. In contrast to the neuromuscular junction, instead of breaking down and destroying the neurotransmitter, if we follow this molecule, it'll bind here, it'll bind here, and then it'll be grabbed by this transporter protein. And what this transporter protein is going to grab the neurotransmitter and then bring it back into the terminal. When we clear all the neurotransmitter from the synaptic cleft, then obviously these receptors are no longer activated and our effect is gone. Okay, so monoamines don't break it down, right? They use transporters. Now there's two main synapses we're gonna talk about, or will have talked about: dopamine and serotonin. Okay. This transporter, here's one thing about biopsych that actually makes sense. The word in front of the transporter tells you what it carries. Okay, so if we're at a dopamine, if this is a dopamine synapse, we'll have the DAT, which stands for dopamine transporter. If we're at a serotonin synapse, we'll have the SERT, or the serotonin transporter. Now, once we're back here in the terminal, a couple things can happen, and this is not short term, okay? This is longer term. We can recycle, or sometimes it does get broken down. Okay, this is where monoamine oxidase comes into play. Okay, now this doesn't do anything in the synaptic cleft. It's not how the signal's terminated. Okay, it's by these transporters. Once we're back here, sometimes it gets repackaged, sometimes it gets broken down. Okay? With some of the early antidepressant drugs, what they do is block monoamine oxidase. If we block monoamine oxidase, that means there's more neurotransmitter to get repackaged and it increases levels. All right, there's some, we don't use MAO inhibitors very much anymore, or you try to avoid them because there's some nasty side effects. All right, so that's how the monoamine, everything's the same until we terminate the signal. Drugs like cocaine, okay, they block the dopamine transporter. Okay, so in this case, if a person has lots of cocaine, the action potential comes down the terminal, voltage gated calcium channels open, calcium comes in, cascade of biochemical reactions causes the synaptic vesicles containing dopamine to fuse and release neurotransmitter. Dopamine will then bind to the receptors and affect the postsynaptic cell. Now, normally, these dopamine molecules will get taken back up by the transporter, but with cocaine, that doesn't happen. These transporters don't work anymore. So what ends up happening is the dopamine stays in the synapse longer. It'll have a larger, more prolonged postsynaptic effect. Eventually, over time, it will diffuse out of the synapse and it'll get picked up by astrocytes, for example. Okay, but it takes a long time for the neurotransmitter to clear. Okay, other drugs will directly stimulate the dopamine receptors. Other drugs like amphetamine. Amphetamine actually reverses the transporter. Okay, so with something like cocaine, if my neuron isn't releasing dopamine, no dopamine is being released. Cocaine will have no effect. Amphetamine, however, if I have dopamine floating around here under the influence of amphetamine, amphetamine will actually cause the transporter to grab this and bring it out into the synaptic cleft. Okay, so when the transporter is grabbing neurotransmitter from inside and bringing it outside, right, we are causing neurotransmitter to be present when it wouldn't normally be present. And obviously, if the transporter is going from in to out, reuptake doesn't occur. Okay, so amphetamine does what cocaine does, plus a little bit extra. All right, so those are the basics of the synapse of neurotransmitters. Everything I kind of went over would be real good for you to know. Understanding how it works will be helpful for you too, because again, I'm going to ask you questions like I did. What would happen if we increase the amount of transporters at the synapse? Okay, so if we increase the number of dopamine transporters, what would happen? Well, dopamine would still get released just fine, right? Nothing wrong with the receptors, so we'd still have an effect. But with more transporters, the neurotransmitter is going to get picked up more quickly, which means it'll have a shorter effect. So things like that will be good to know for the final. I'm going to pause the recording